Thank you all for joining this webinar. I'm Jaya Krishnan, co-founder of Genome Biologics, and today I'm excited to share with you one of our latest technologies in cardiac disease modeling. This is based on a cardiac organoid platform that we have established and used for drug discovery and cardiotoxicity testing. So today, I'll now start with the background to TrueCardium, how we developed it and how it functions, and then move on to disease modeling and end with some of our successes in using this platform. Now, to start with, our organoid platform, our human cardiac organoid platform, is based entirely on human iPSC cells, and this is then differentiated in a dish to generate this mature organoid that we're going to talk about. Now, this is an entirely self-organized process. We do not add additional cell types or exogenous cell lines to this culture. Rather, starting from this initial iPSC population, we are able to generate this entire plethora of cells just by controlling the growth factors that are added and the time point at which they are added. So our organoids comprise of all the cell types of the native human myocardium. This includes not just the cardiomyocytes, but also the neurons, endothelial cells, fibroblasts, macrophages, and of course the mural cells that cover the blood vessels, the pericytes and smooth muscles. Now, unlike what is commonly available when talking about cardiac organoids, ours are unique in that they contain all of the native cell types. And what this enables our organoids to do is to also self-organize to exhibit the cell organization, the tissue structure, and cellular organization within this tissue. Typically, organoids only contain just a fraction of these cells. Some contain only cardiomyocytes, or some organoids may contain cardiomyocytes, endothelial cells, and fibroblasts, or just cardiomyocytes and fibroblasts. It is our view that these other platforms may not accurately recapitulate the complex organization that is the heart. So to move on from here, I would like to just share with you as to how this organoid is developed. We start with a two-dimensional culture of human iPSC cells. Now, we then dissociate these cells and transfer them into a hanging drop or place them into a low attachment well. The point of this is to use gravity to enable the cells to settle and form a cell ball as such. Now, it is on this cell ball that we apply our 7 waves of growth factor cocktail to then enable us to develop all of the different lineages that are seen here. So, for instance, in the course of development, what you start seeing is the initiation of neuronal cell development. So you start with the development of cardiomyocytes, as stained in pink, representing alpha-actinin, a cardiomyocyte-specific marker. And with development comes the emergence of the TUJ1 neuronal population and of the ULEX endothelial cell population. And what you see here is that in the course of the development process, we have expansion of these endothelial and neuronal networks, and also that of the cardiomyocyte population. These organoids grow up to a diameter of about 400 to 600 microns when fully mature at day 35. At day 0, when the organoids first appear, we then add a further maturation period to then enable them to form these adult organoid structures. Now, these adult organoid structures, I will show you in the next slide, recapitulate the microscopic features of the human adult heart. But before we go there, I would just like to point out that by performing RNA sequencing on organoids at the various stages of development, we are also able to correlate them with specific time points in mouse heart development and through extrapolation to human heart development. A reference data set exists for mouse heart development, but not for human heart development. Thus, we matched our human organoid our human organoid RNA sequencing to the mouse data set. And when comparing to the mouse data set, what you can already see is that our earliest organoids exhibit the features of an E8.5 embryonic mouse heart, extrapolating to about 4.5 weeks of human heart gestation, human heart development. Now, as we progress through the different time points, what we then come to is the postnatal heart, a P1 heart, and when we let the maturation protocol progress, we then culminate with the mature adult heart, which correlates well, according to Pearson's correlation analysis, with that of the adult heart. Now, I should mention that these correlation scores are based on comparisons to mouse. When we compare specific data points to available human heart data, we actually get a higher correlation value, in part because our starting material is in fact human cardiac organoids, thus it matches better with human cardiac tissue. So some of these correlation are shown in this RNA-seq heat map below. Now, when we then go on to performing single-cell sequencing, what we then start to see is that indeed we have all of these different cell types. So these are the cell types here as quantified from staining. And below you will see the RNA sequencing data, where we see a lot of the populations that are present in the human heart. Now, this includes neurons, as you see here. This we have marked using TUJ1 staining, and these are blood vessels. Now, you may note that the density of these blood vessels is relatively high, as one would expect, but more so, you may see the three-dimensional nature of these blood vessels, suggestive of the presence of a lumen. So we in fact know that we have perfusion within these organoids, and we also know that due to the presence of the blood vessels, we are able to also test the efficacy of, say, larger compounds or biologics, which typically cannot enter an organoid by diffusion alone. But due to the presence of our blood vessel system, we are able to deliver organoids not just superficially to the, we are able to deliver biologics not just superficially to the organoid surface, but also internally. And this is brought about by the blood vessel system that is present here. Now, not only do we have blood vessels, but you may see from the picture below that we have mature blood vessels. So in larger vessels, we have smooth muscle cell coverage as marked by red by smooth muscle actin staining. And in the capillaries, not shown here, we see the presence of parasite coverage. So we know that the blood vessels in our system are stable and mature. And this is important because immature blood vessels are largely non-functional and tend to serve a different purpose. This purpose is not compatible for normal heart function. Thus, the presence of this mature endothelial network is again characteristic of the native human heart. Now, when we start taking slices of our organoids, what we see is that, as you might notice in the reference cross-sectional picture from a native human heart, the cardiomyocytes, here marked in green, blue being DAPI, the cardiomyocytes exhibit the expected rod-like shape that is typical of mature cardiomyocytes and not typical of immature embryonic or early postnatal cardiomyocytes. This is important because this difference in cell shape is a reflection of the change in the transcriptome of these different cell types, and also a change in its metabolic profile, change in contractility, etc., etc. Not only do we have this rod-shaped structure, but our cardiomyocytes also exhibit polarization. And this is, of course, very important for synchronized force transduction in the native heart, and in our case, in the organoids. Below, you will see just staining for the sarcomere with sarcomeric alpha-actinin, and you will see that the sarcomeres look normal and also exhibit polarization. Now, when we delve a bit deeper in and ask whether the metabolic profile also changes between the immature day 0 organoid with the adult type day 35 organoid, as one would predict it would, we in fact see this shift. So in the day zero, we see metabolites typical of highly proliferative tissue, and this goes down slowly in the day 35 organoids, and in reverse. At day 35, we see a lot of metabolites, more typical of the immature stage, and more typical of the immature stage, which again are downregulated when you come to the day 35 mature cardiac organoid. Now, with the change in metabolism, between day 0 and day 35 comes a change in contractility. Now, this metabolomics profile, what it also shows is an increase in the utilization of glycolysis in the early day 0 heart, and an increase in oxidative phosphorylation type metabolism in the day 35 heart. And this is important because as it is in humans, the embryonic heart is largely glycolytic due to limited access to oxygen. However, after birth, the excess is increased, and this then leads to a shift to a predominant mode of oxidative type substrate metabolism. Now, this is also important in terms of cellular energetics. The embryonic heart does not require as much power as that of the adult heart due to the smaller size of the organism. However, as the adult ages and matures, the heart is subjected to greater stress, and there's a greater need for efficient, strong function in order to pump blood throughout the body and to the peripheries. This is reflected in the change in the mode of contractility, as shown here. In the day 35 organoids, in part due to the utilization of OXPHOS processes that produce more ATP per unit time, we see an increase in the contractile amplitude. This is reflective of the contractile power of the heart. And this happens concomitant with a reduction in contractile frequency. So essentially, the heart is so efficient, the adult heart is so efficient that it doesn't need to pump frequently in order to achieve its aim of distributing blood throughout the body. In contrast, the embryonic heart functions at a lower amplitude, but at a higher rate of frequency. So it works a lot harder because it cannot generate as much power per unit time. And this is again typical of the difference between early embryonic or postnatal hearts with that of the mature myocardium. And these data are quantified in the graph below. Next, we asked the question how these organoids would respond to compounds of known functionality. So, for instance, we stressed these organoids with phenylephrine and carbachol and performed calcium flux analysis. Now, changes in alterations in calcium flux underlies cardiac contractility. So we are able to measure changes in contractility by following the flux of calcium. And as you note here, stimulation with phenylephrine increased the frequency of beating, while carbachol, as one would expect, decreases the contractile frequency. We then performed ion optics. This is more a physical and visual measure of contractility, wherein we are able to directly see changes in the contraction and constriction and elongation of the sarcomere in real time. Now, with this assay, again, using well-established drugs, as shown here, we see the expected change in frequency, the expected increase and decrease in frequency in response to cytosporin, and an almost complete loss in contraction in response to doxorubicin. So it should be noted that doxorubicin is a highly cardiotoxic compound, and at the doses used here, we see an almost total loss in contractility. But of course, at other doses, we are able to titrate the effects of this compound. So next, what we did was we performed a screen. The point of this screen was to address how physiologically relevant or clinically relevant our organoids might be in predicting the cardiotoxic or safe nature of specific compounds. So rather than starting fresh, what we did was we relied on a panel of compounds known as the SEPA panel. This is a very well-established panel of drugs whose effects in clinics, in mice, in vitro are very well established. So using this platform, we then performed a mini screen with these SEPA compounds on our cardiac organoids. And as you see here, we had a large degree of concordance between the expected cardioprotective nature of compounds and also with the expected cardiotoxic nature of compounds. So we have almost a 95% rate of accuracy. I should also mention that our organoids have been distributed quite widely. We now have about 15 plus publications using our organoids from various groups. So it is not just us that has worked with this, but we have also worked with these organoids with our partners and in collaboration with other entities who have directly tested these organoids in-house in their hands. So we think our system is quite robust, amenable to transport, and also amenable to manipulation, not necessarily by experts. It's fairly easy to work with. Now to the exciting part. I would like to talk about the disease models that we have developed using this organoid platform. So we have developed a few such models, but I won't go into all of them in the interest of time. I will start with inflammation. This is something that is very topical of late. And an example of an inflammatory project that we have done is a project on cardio-inflammatory CHIP. So CHIP stands for clonal hematopoiesis of indeterminate potential. CHIP is usually associated with leukemia, a good example being acute myeloid leukemia. Leukemias, however, tend to be early onset indications, whereas cardioinflammatory CHIP is a late onset indication, which affects males and females at comparable rates. And happens 50 in subjects 55 years and above. Now, what this indication comprises of is the establishment of a mutation in the monocyte macrophage population of cells. The two most common mutations are TET2 and mutations in DNMT3A. And these mutations tend to increase the infiltrative capacity of monocytes and macrophages, and more so increase the inflammatory induction within the target tissue. And this then, as a consequence, leads to cell death and fibrosis. So what we tried to do was we tried to recapitulate this whole indication in our organoids. And to do so, we co-cultured our healthy organoids with macrophages bearing a TET2 gene knockdown. Now, the TET2 mutation associated with cardioinflammatory CHIP is a loss of function mutation. So we can, to an extent, mimic it by knocking down TET2, thus reducing its functionality. So in doing this, we also labeled both the control macrophages and the TET2 mutant macrophages with a cytotracker dye in order to enable us to easily follow the path of these exogenous macrophages. And what we then tried to look at was the infiltration of these cells, the capacity to induce inflammation, and downstream consequences of that inflammation. So to start off with, what you can see here is a nice infiltration of the macrophage Q-tracker labeled macrophages. This is the cell dye, Q-tracker, the Q-tracker labeled macrophages into the organoid proper. This infiltration is not just at the surface area, but rather deep within the organoid itself. I should mention that our organoids do not have a necrotic core. This is because of the vascularization and the perfusion that is present within the organoids itself. And thus far, we have grown them up to 9 months. They get bigger, but they do not still develop a necrotic core. So that is why we can quite easily and readily image and characterize this deep infiltration into organoid proper, where typically you might expect to find a necrotic core. Now, coming back to the story, what you see here is that when we have effective knockdown of TET2 using our siRNAs, and just by inactivating TET2 in these macrophages, we immediately see an increase in the infiltrative capacity of these macrophages, as shown in 2D, and better reflected in 3D. So what you can see is an increase in the intensity of the pink staining here and of the pink staining above as well, in organoids co-cultured specifically with the TET2 deficient macrophages. And the quantification is performed here, wherein you see both an increase in local and global coverage area by these Q-tracker labeled macrophages, and confirmed by qPCR. So what we did here was we took the organoids, washed them to remove any cells that might be sticking weakly, and then we subjected them to qPCR analysis, and we do indeed see an elevation in markers of macrophages. Next, we then ask the question, does this infiltration lead to an increase in a cytokine burst or cytokine induction, as is observed in human patients? And that's indeed what we see. So using a Luminex assay panel, what we can see is an elevation in IL-1 beta, IL-6, and IL-18. These cytokines are similarly elevated in patients, in cardioinflammatory CHIP patients, and also an increase in the RNA level of these and other cytokines. So next we ask whether this increase in microenvironment inflammation within this tissue would have an impact on on cardiomyocyte viability. So in patients, it's been noted that the increase in this whole inflammatory induction leads to increased cardiomyocyte cell death, downstream of that being fibrosis, heart stiffening, and cardiac dysfunction. So taking that whole series of expected steps, we looked at cleaved caspase and co-stained with sarcomeric alpha-actinin. So the overlap between cleaved caspase and sarcomeric alpha-actinin is indicative of specific cardiomyocyte cell death. So we do indeed see an increase in apoptotic staining in the cardioinflammatory CHIP organoid model, and this is quantified here. Also confirmed using an independent measure with cardiac troponin T. Now, cardiac troponin T is an assay to look at troponin T released by the organoid into the conditioned media. This is similar to a clinical test that is done to look at potential heart damage using the high sensitive cardiac troponin T assay. So the basis is essentially the same. And so by looking at levels of CT and T in the conditioned media, we can have a rough estimate of the extent of cell death. And as you see here, there is indeed an elevation of cardiomyocyte cell death consistent with our staining quantification data. If we then look at the RNA level, we also see the elevation in apoptotic drivers and also of caspase components. So the next step being, is there fibrosis? So when we perform collagen 1A1 staining, this is a fibrotic marker staining, we again detect an increase in fibrosis in the cardioinflammatory CHIP organoids, whilst little fibrosis was noted in control organoids. And this is again reflected in the RNA profile here. Now, with all of these changes, the apoptosis, the fibrosis, The downstream consequence, as I mentioned, would be a change in the contractility of these organoids. So we asked the question, do we also detect this change in cardiac function? And indeed we do. When we compare the control organoids with the cardioinflammatory chip organoids, we see a decrease in contractile amplitude and an increase in frequency. So essentially, these hearts are less functional, and with time, the function will decrease even greater. And this is a consequence of the upstream effects on the cardiomyocyte cell death and fibrosis. And these data are quantified here using calcium flux to look at frequency of contraction and the amplitude of contraction, wherein the amplitude goes down and the frequency increases. So very quickly, I'd like to tell you about our other models. So CHIP is not the only model we have. In fact, it's one of our more complex models. We also have models comprising of LPS and other immunogenic inducers. But I should also mention that we have done similarly complex projects, not just with macrophage co-culture, but also with B-cell, T-cell co-culture, and also with in combination with chemical compounds or biologics. So we are able to look at different inflammatory processes or situations. So next, I'd like to go to myocardial infarction. This is a very well-established stress with broad human health effects. And essentially, what happens here is due to an occlusion of a vessel, an area of the heart has limited access to oxygen, and this leads to cardiomyocyte cell death. Cardiomyocytes, like the neurons, are highly dependent on oxygen for its function and are also very dependent on oxygen for its survival, and thus it is very quickly affected in response to severe hypoxia, such as seen in myocardial infarction. So when we tried to model it in our organoids, what we did was we subjected the organoids to 1% oxygen. This is severe hypoxia, and we then looked at its oxygenation capacity. And what you see here is that there is very limited oxygen uptake by these cells, and as one would expect due to its hypoxic nature. And we also see a decrease in viability in these cells. Concomitantly, we also see an increase in cardiomyocyte cell death, as reflected by the elevated levels of cardiac troponin T in the conditioned media of these organoids. So these are all changes that are also observed in human patients. Now, we then ask, do we also see the increase in cytokine profile typical of infarction? And we do. So as noticed, as seen here, we see the elevation of interleukin-1, interleukin-18, and interleukin-6. And also, this directly links to an elevation in fibrotic markers such as COL3A1, MMP, and also fibrotic drivers such as TGF-beta1. Finally, we also then looked at hypertrophic markers. We see an elevation of NPPA and NPPB, and also MYH6 and MYH7. These changes are again similar to those observed in human myocardial infarction patient biopsies. And in this system, as one would expect, due to the death of cardiomyocytes and the increase in fibrosis, we have a decrease in cardiac amplitude, so decrease in contractile power, and an increase in frequency. Next, I'd like to go into cardiotoxicity, where we have performed various screens to identify the toxic or safe nature of compounds, and also we have performed this screen in combination with compounds in order to identify drugs that may alleviate the effects of anthracycline cardiotoxicity. Now, cardiotoxicity, now anthracyclines are a commonly used chemotherapeutic agent used in clinics mostly for the treatment of breast cancer and certain leukemias, but they also have a very negative impact on the heart. So what we did was we asked the question, how does doxorubicin affect our organoids? What you're looking at here is a viability readout and all cell viability readout. So we do not know who exactly dies here, but we do in fact see that with increasing doses of doxorubicin, there's a significant drop in viability. If you look specifically at cardiomyocyte cell death using cardiac troponin T in conditioned media to get a rough measure of this, we can see an increase in cardiac troponin T in conditioned media, indicative of increased cardiomyocyte cell death, as one would expect from doxorubicin treatment. What we also see is a metabolic shift towards glycolysis by the increased levels of lactate and significantly compromised contractility with the loss of beating here. Now, what we did as well was we asked the question, we seem to readily detect the effects of cardiotoxicity. Can we also distinguish toxic compounds from safe compounds? So what we did in order to ask this question was we did a blinded retrospective study on a subset of compounds that are known to be safe and also on a subset of compounds that were cardiotoxic either at phase that were cardiotoxic and were stopped at later stages of clinical development. And what you see here is, to a large extent, our organoids are able to predict the safe or non-safe status of compounds. We did get some false positives, but we can discuss later why we think this is so. Now, this is important. We think that our organoid platform is able to better predict the toxic or safe nature of compounds, in part due to the fact that it has all of the cell types relevant for human heart function. So if one were to perform such a screen on cardiomyocytes, all of these compounds may appear safe. But if some of these compounds were to have a negative impact on neurons, this would not be seen in a cardiomyocyte only or a cardiomyocyte fibroblast only organoid or 2D system. So using such systems, you would essentially be blinded to the effects of the negative effects of these compounds on other cell types. But as our organoids contain all of the relevant cell types, we are able to detect negative effects. So if a drug were to negatively affect neuronal growth or neuronal survival, this would have a direct effect on cardiomyocyte function and survival. And because we have this interaction and this system established within our organoids, we are able to, we think that's the reason why we are better able to predict toxicity or safety. Now, I would like to go into HFpEF. HFpEF is also a very important indication, more so in our current age of overnutrition. This is a metabolic disorder which is characterized by heart failure, but with the preservation of ejection fraction. Ejection fraction essentially relates to cardiac amplitude. Now, this is the opposite of HFrEF, heart failure with reduced ejection fraction, which is basically the myocardial infarction and heart failure data that I showed just before. So in the context of HFpEF, one of the key features is the presence of lipid droplets within these organoids, within HFpEF cells. This is due to the lack of oxidative metabolic function in these cells, leading to the accumulation of lipid droplets. This is something previously observed in patients as well. Sorry. In our HFpEF organoid model, we first and foremost do see this increase in lipid accumulation. We also do not detect a change in amplitude, whilst we do detect a slight increase in frequency, again, as is typical of the human population. And more importantly, when we look at the molecular characteristics, what we see is that we detect an increase in fibrosis as marked by increased ACTA1-2 called endocollagens, and also by an increase in the fibrotic driver TGF-beta. We also have in our hands an antifibrotic agent, which when applied simultaneously in HFpEF organoids leads to a reduction in fibrosis, Now, as you can see on the patient data here on the right, this is characteristic again of HFpEF. With regards to the elevation of fibrosis in the tissue. Now, I did not show you this data, but typically in myocardial infarction samples, due to the localized nature of the stress, you tend to have large foci of fibrotic cells. This is because in response to an occluded blood vessel, it is mostly the tissue around that blood vessel that is affected, leading to this very focal pattern of cell death and of fibrosis. However, in HFpEF, this is more diffused because this is more of an tissue environment type indication as a result of altered systemic metabolism. And due to that, you also see a more diffused staining of fibrotic markers such as COL1A, as shown here, and also as reflected in the sections on the right. And again, with the antifibrotic agent, we're able to reduce the extent of fibrosis. Now, moving on, we also see that expression of hypertrophic markers such as NPPA, ANP, or NPPB, BNP is elevated and also brought down with addition of the antifibrotic agent. And the same happens with respect to inflammatory markers, IL-1B and IL-6, again, consistent with the clinical phenotype. Bob, I'm sorry, just give me a few minutes. I'd just like to end by talking briefly about a congenital disease model that we have established. I would like to highlight the fact that, here we are talking about a specific mitochondrial myopathy, Kearns-Sayre syndrome, but this is to convey that we are able to happily work with congenital models or genetic disease models. Now, Kearns-Sayre syndrome is a fairly complex indication characterized by 5 kb deletion of the mitochondria. So this then leads to a fairly complex phenotype, in part due to the heteroplasmy that can be present in mitochondrial populations. But just to talk about the phenotype itself, so what we know is that due to this deletion, we have for the most part comparable expression with the controls, but due to specific proteins being misexpressed or expressed at lower levels, what we do not get are the supercomplexes needed for normal mitochondrial function. This is most relevant for complex 1 and complex 4. In addition, we also see changes in proteins involved in maintenance of mitochondrial cristae activity. This then leads in part to these dilated type mitochondria that are highly enlarged compared to normal mitochondria as seen in controls. And this mitochondrial defect then of course lead to altered oxygen consumption and stark changes in its ability to perform OXPHOS, oxidative phosphorylation, and also is reflected in the organization of the mitochondria. Typically, mitochondria are organized as shown here. They span the length of the cardiomyocyte and alternate with sarcomeric alpha-actinin staining. So they are in fact almost striated the same way the sarcomeres are, and you can maybe see some of the striations here. However, in the mutant, this striated pattern is completely lost, and they form very strong foci surrounding the nucleus mostly. And this is better reflected here, where you see the almost perfect overlap of mitochondrial ND3 staining with that of sarcomeric alpha-actinin, but ND3 being one of the deleted proteins, when we then stain with ND3 in KSS, one of the deleted genes in KSS, then we see the loss of this perfect organization and more of a dispersed type phenotype. You do not see all of the cell staining for ND3 because, of course, the mutant mitochondria would not express ND3. This links to the whole heteroplasmic effect of this indication. In this mitochondrial dysfunction then leads to a decreased consumption of oxygen, decreased ATP generation, and significantly retarded contraction. So as noticed here, there is a significant drop in the cardiac amplitude and a huge increase in the frequency. And this then, of course, leads to the elevation of ANP, a strong hypertrophic marker leading to many processes that leads to remodeling of the of heart tissue. I'd like to end very quickly just to let you know that we have successfully developed in-house and with partners a number of assets using our platform. We have also won an EIC grant on the basis of data from these organoid systems. And I would also like to say that REPROCELL has been extremely kind and helpful to us, and they have provided us with some of the allogenic and the clinical cell lines as shown here, and also some of their research use iPSC lines. We have integrated this into our organoid workflow, and we can generate wonderful organoids using their organoids. So this has been very useful to us to expand our panel of organoids, and also to let you know that we are able to work with a diverse range of organoids, be it allogenic or genetically mutated. With that, I would like to thank you.